Hallo zusammen, herzlich willkommen, welcome and greetings from Soapbox Science Düsseldorf, Germany. I hope every one of you is keeping safe and staying put. So today we are gathered here to present some of the leading scientists in Düsseldorf and the areas of Germany, and they will be telling about their research. So before I begin, I am Tanvi Khera. I'm a virologist here in Germany, and together with my colleague Audrey, we will be hosting the English edition of this session. The event is actually organized into two different parts. The first one is the English session that will last up to 12:30, and the next session after a short break will resume with the German session beginning at around 2 p.m. So fine, let's begin. So what do you know about Soapbox actually? So I will give you a little brief history about the Soapbox. The Soapbox dates back to a long time when people in the UK used to promote their values or about religious or political views in a form of a speaker's corner in Hyde Park. And from there, we got this idea of having a soapbox, but with the promotion or with the value of promoting women in science. So with this in mind, we started this a decade ago and promoted Soapbox Science. It has been in various cities, and for the first time in Düsseldorf, we are holding this event here in our city here in 2020. So what exactly are we promoting is basically that there is the visibility of female scientists in science, and we really want to put focus on that. And in traditional ways, the Soapbox event would have been in a public area, engaging the public and making it more for the kids, for everyone. You don't have to be from a science background. But due to the recent current pandemic, we have moved to an online streaming platform, and there we are. So if wherever you are, just drop in a hi to say so that we know where you are streaming from. All right, so when we talk about science, what usually comes to your mind? So when we talk about science, a lot of you might be engaging or might be relating it to Big Bang Theory or even Eureka or even Doctor Who. But trust me, it's much more than that. Especially during these times, scientists and also science has gained a lot of visibility as well as appreciation during the COVID times. And trust me, there is much more to that. So that's why we are here gathered, and we've put together a bunch of scientists, female scientists for you, who are passionate and who wants to share their research with you. So the pandemic has really taught us about not just about the infectious disease, but also about environmental hazards, but also on the mental awareness situations. So we tried to put together all the scientists from different scientific background, and we are going to present them first. So to begin with, we have our first speaker, Ms. Chiara Bruno, who is engineer specialized in renewable energy, and her mission is to help reduce CO2 emission and promote a more sustainable way of living. So I present to you here, Chiara.
Thank you very much to all the Soapbox Science Düsseldorf team for organizing this event and giving me the possibility to be here today and to introduce a little bit of what my work is. And I really hope you guys will enjoy it. So yeah, as Tanvi said, I am a renewable energy engineer. I studied in Italy, in Rome, where I come from. I studied renewable energies, and then I specialized myself in sustainable energy in Stockholm, in Sweden. So a little bit of background about my job and my research. What is engineering? For me, engineering is the application of science to reality. And for me, this is very important because as much as I enjoy learning about science, I really enjoy the fact that science can be actually applicable to real life. And energy, why did I choose energy? I think energy is really what we need in the world. It's like we need in our day-to-day life. It's what our body does. Our body basically converts the energy we get from the food to the energy we use for moving and for living. But also, anything you can think about in your life needs energy. For example, if you need to charge your laptop, if you need to charge your mobile, if you need to heat up your home in winter, you need energy. So you need heat and you need electricity. And what is renewable? So why renewable energy? Renewable is everything that can actually be renovated after we use it. So for example, if we think about sun, if we think about wind, if we use the energy from the sun today, the sun will still be there tomorrow for us, so the energy will be there. But if we think about, for example, coal, if we use coal, well, tomorrow we're not going to have the same amount of coal because it takes like millions of years to produce this coal. So coal is not a renewable source. And then I would like also to speak about sustainability because it's a little bit different. So what is sustainability? Sustainability is our ability to exist on the planet like in the same way we do today, the same day we can do it tomorrow. So to make sure that our lives and our environment will be kept preserved in the next years. And everything that is destroying this is not sustainable. So exactly, for example, CO2 emission and greenhouse gases emissions are altering our world and altering the atmosphere or the earth, and this cause climate change, and this cause a lot of changes in the nature, meaning that basically if we go on living like we live today, we will not have the same Earth, will not have the same planet in 1,000 years, probably. We are going to destroy this planet. So this is why I got really passionate about this topic, and this is why I decided to study this and then to work into this field. Yeah. So basically, to give you a bit of background also in numbers, because I'm an engineer, we're talking here about 2/3 of CO2 emissions coming from fossil fuels. So coming from plants that produce energy that we use every day. So we want to basically reduce this amount of fossil fuel that are used. And we can take actions in our everyday life. We can try to use less energy. We can try to buy from renewable sources. We can try to also like use less plastic, consume less, go to a more vegetarian eating way of life. And I decided also to carry this into my job. So what do I do for job? I'll show you here. I optimize power plants like wind farms. So my job is basically to make the wind farms, and so these little wind turbines, I mean, of course they are bigger than this, to make them more like efficient and also more cheaper so that we can put more of them and have more energy from them. But yeah, so today basically I would like to show you a little bit of facts about wind energy that you don't know, because in Germany it's quite common to have wind energy, so we have still a lot, but the potential can grow. So first fact I would like to show today is the fact that, are you wondering if wind energy is expensive? Well, not really. So as a fact, in the last 10 years, the cost of producing wind turbines and wind energy went down 60%. So that's a lot. And now we are able to build this wind farm in a very, very knowledgeable way. So we have the knowledge to build them, but also a lot of optimization can be done. So basically this is my job. So how can I build this in a even cheaper way, in a more sustainable way, so that these plants are actually good for us, they are not harming us. For example, you could have heard the fact that wind turbines can be harmful for animals. Let's see, birds. Yes, that's true. Sometimes birds, they fly here, and they don't really see the blades because they are rotating quite fast. The speed at the tip of the blade can be up to 90 meters per second, which is very, very fast. And yeah, it can happen that they are hit by a blade. But on these two topics, first of all, unfortunately, every human building is affecting animals, unfortunately. And second of all, we perform always environmental studies on the sites where we want to put the turbines, so that we are sure that if there are protected birds, for example, with them we don't buy the turbines there, we don't build the turbines there. But also we have systems, for example, we can put thermal cameras on the turbines that will detect the birds, and they can either shut off the turbine, so it's not rotating anymore, or they can emit sounds so the birds are scared and they don't go in the turbine. And the same we have for bats, for example. So we can stop the turbine where the bats are more active, for example, they come out often at in the sunset and sunrise, So we just stop the turbines where we know the bats are going to be there. And this, of course, is reducing a lot the possibility that can crash into the turbine. Then, yeah, another topic would be noise. Yeah, I mean, these things are noisy. Yes, they are. Every rotating equipment makes noise, of course. And this is why we don't put turbines in the middle of a city, because they would be too noisy. So you can be assured that actually if they will build a wind farm close to your place, everything has been calculated so that you will not hear this noise. Because the wind farm will be far from your house. But also, we measure noise. We have, the turbines have like noise, low noise emission modes, so they can be more silent, basically. And also, we can shut them off in some directions. So if they, for example, if the wind is coming in this direction and that would make more noise for your house, then we stop it from that direction and we only produce when it's coming from another direction, because the turbine can move in this direction to follow the wind. Yeah, then I would like to talk a little bit about dimensions. That could be interesting. So, you know, the highest turbine in the world is actually at the moment 260 meters tall from bottom to the top. And that means, well, if you are dialing from Germany, probably you know the Dome of Cologne, so the main cathedral, which is 176 meters, I think. So it will be some like here. So, yeah, pretty big. Why do we make them bigger? Simply because they harvest more energy and the efficiency is higher. And also, I would say that the environmental impact would be actually lower. That's because with less turbine, you can produce more energy. So you will see, they will be bigger, but you will see less of them. So that's why I also think it could be, it's very nice if we can just build them bigger but less. Then I also would like to give you a bit of insight about floating wind. So do you know, you probably know that there are also turbines offshore, so in the sea. This is a very new technology that we are developing, and it's just basically, instead of having a monopile, like a really long base which is gone into the sea until the seabed, you would just have some kind of a steel structure which is floating on the water. And then the turbine is just on top of this steel structure, which will be anchored to the seabed like a normal ship, basically. And this will allow us to exploit way more sites, because we can go farther, like out in the sea, and we can go where the water is more deep. And also, yeah, basically where the water is deeper, and means the wind is also stronger, because farther you go from the shore and the stronger the wind, and also you don't really impact the visibility, or you don't impact the environment, landscape, let's say. Another cool thing about offshore that I recently learned in my research is that the turbines are actually not harming the environment, but since it's forbidden to fish where the offshore parks are, basically at the bottom of the turbine, you create some kind of artificial reef, some kind of artificial environment for the fish, and actually the biodiversity is increasing, because fish go there to live. They are not bothered by fishermen, because fishermen cannot fish them there. So actually we saw like this increase in biodiversity in the wind farms, which was kind of very, very nice. And this last topic I would like to mention is the fact that, yes, probably you're wondering if renewable sources are enough to sustain us. The answer is probably not, but this is why we have to develop all of these resources. So the wind, the solar, but also we have to couple them, for example, with storage systems, which could be as simple as batteries, like the battery you have in your cell phone, to something a bit more, let's say, complicated, like water storage. So you can basically use the excess of energy that you have sometimes to pump up water from like a lower level to an upper level, and then basically when you need that energy, the water flows down, and with a hydro turbine, you can produce more energy. So basically, yeah, you couple the wind farms with some other sources, and then we want to secure basically the energy provision all the time. Another way, for example, would be to use the excess energy, because if you put a lot of wind turbines, they all go at the same time. Maybe you have too much energy, but you can store it, for example, with hydrogen. You can produce hydrogen with the electricity you have in excess from the turbine, and you can use this hydrogen later for any other energy purpose that you might have. So there are many, many different types of hybrid system we can think about there, and this really needs a lot of research and a lot more of optimization to make it economically viable. So that's basically my job, to always try to find the right way so we can put more renewable energy and be more sustainable. I hope I was in the time.
Yes, you are. Grazie mille, Chiara. That was really, really, really nice. Thank you so much. So we have some questions for you.
Oh,
and
okay.
the question is, yeah, for sure, of course. So someone asked that what nowadays is the production cost from the wind, actually? Like to produce power, what is the production cost from wind these days?
So I would say that this really depends on the country. This is because, of course, like the cost of, for example, permitting the land, the cost of operating the wind farm, it's really dependent on the country. But I would say that at the moment, the cost that we have to produce energy from the wind in some countries is even lower than the cost that we have to produce energy from turbines. So we are talking about a range that goes between like $30 per megawatt hour produced to like 60 euros per megawatt hour produced. It really depends on where you are doing this, but this cost is as foreseen to be declining in the next year, and even get the wind more competitive than coal and fossil fuels.
So someone was actually also questioning about that how is the energy transport happens from the offshore turbines to the land. Yes. So what are the different ways? I think it's very interesting, right?
Yes, exactly. That's one of the main challenges, because as I said before, the offshore turbines can be quite far from the shore. So there is a long distance that this electricity has to basically to travel to reach the substation. So there is a substation onshore, which is a place where the electricity is transferred from the offshore to the onshore grid. And basically, the electricity is transported by a direct current. So direct current is a type of current that transports electricity in having less losses than the alternate current. The normal current that we have here, it's alternate, but we have direct current cables that transport this energy from the turbines offshore. So really, really big cables, basically, that goes below the sea, and they transport the energy up to the shore.
And Chiara, finally, if you could just very briefly, in like 2 lines, tell us about, because the United Nations Sustainable Development Goal by 2030 is to have a more sustainable living. So how, from your field, are you going to, sort of translate this? And what do you kind of perceive or see that in 2030, how is it going to be?
Well, I would say that, first of all, I love the fact that my company wants to grow a lot. And I would really encourage, if I could, like the government to give us even more subsidies, because in some governments we are actually lacking subsidies now, but they are still needed. So also if Germany, for example, put subsidies to phase out the coal, I think that's very important, because then we can really focus on this renewable energy. Something we can do, like in our daily life, is of course taking small actions. For example, in winter we can heat up our apartment 2 degrees less, and this is already a big impact for the environment. We can choose to buy from renewable energy electricity providers. This might be a little bit more expensive, but it's very good for the environment. And we can, in general, I think that really everybody in our day-to-day life can make small actions to make the world a better place.
Perfect. Very nice.
Thanks a lot.
All right, so moving on. What do we have next? So we have from an engineer, we move on to an immunologist. So we present to you Ms. Panayota Marmirelli, or Naya. Maybe I call her Naya, so we all can just refer her to as Naya. So Naya is actually an immunologist with the focus on autoimmune diseases. She's currently doing her postdoctoral research at the Medicine University in Mainz. And before this, she has been involved in various science communication activities. For example, she was the recipient of the first prize at the FAME Lab in the Lower Saxony and was also awarded to be present at the German level. So I present to you Naya, who actually presented her science communication in only 3 minutes. But today, I'm hoping to hear more, Naya. Okay. The stage is yours.
Good morning, Tanvi, and thanks a lot for allowing me to be part of Soapbox Science in Düsseldorf. So as she kindly introduced me, I am working with our immune system and how its actions affect autoimmune diseases. So my main interest is on T cells. These are small cells of our immune system that are mostly important in order to defend our bodies against pathogens, microbes that are going to harm ourselves, or even chemicals. So there are several categories that mostly we can divide them into two major so that it's more easy for you to understand. And I chose some colors so that it's easier also to remember them. So the ones that are actually important in order to defend ourselves from bad agents, microbes, chemicals, allergens, and whatever, they're called effector T cells. And yeah, as red color is usually a little bit more aggressive, let's say, I chose this ball just to resemble them. So these are very important. Once a foreign agent comes to our body, they have to recognize it, activate, and start fighting it. On the other side, when these guys are no longer needed because the bacteria or the virus is eliminated, we have some diplomats in our immune system, those so-called anti-inflammatory cells, that tell our effector cells to shut down their action, their weapons. So it is really important, those two guys, to stay in a balance because you don't want way too many of them when there is nothing wrong trying to enter our body. But on the other side, when you have an infection, you don't want these guys to be so many that they could just stop them. You really need to find a way in order to balance them. There are other examples, like for example in tumors, you don't have enough of the effector cells in order to fight the tumor cells. You have a lot of the anti-inflammatory, and this causes a lot of troubles to the treatment of cancer patients. Same thing happens also in autoimmunity, where you have a lot of effector cells actually targeting your own system. It's like a civil war that needs to be shut down. So what in my projects I'm trying to do, I'm trying to find ways in order to balance these guys here, either to promote more of the effector cells when you need to fight a persisting infection, or to limit them by inducing more of the diplomats of our immune system in order to reduce the inflammation when it's no longer needed or when it's unwanted. In the cases of autoimmunity, like people suffering from inflammatory bowel disease or from rheumatoid arthritis, or even in the cases of allergy and other unwanted autoinflammatory conditions. Someone would ask how we can do it. There are a lot of drugs out there that do this job, however they're not specific. They act not only on our immune system, but at the same time they also act on other cells of our body. In my lab, we are trying to find natural compounds that could actually influence this balance without having severe side effects. And upon a screening of natural metabolites from a bacterium from the soil, from the earth, I don't know how I always miss the German word for that. We came up with one compound called zorafen that can actually do this job. Zorafen actually tells the cells to change their appetite. So here we come back to energy, just like our previous speaker talked, but more about the energy of the individual cells now. The effector cells are mostly relying on a lot of fat. They need fat in order to be able to fight the pathogens, to proliferate and become more, because you need more soldiers when there is an infection going on. And in order to stop them, what we do is that this compound actually starves them from fats. It prevents them from being able to eat fats and use them. On the other side, these anti-inflammatory diplomats don't really rely on fat. And since both of them take from the environment the same signals, this energy availability is really a key that can control this balance. So Zorafen can inhibit our effector inflammatory cells, but at the same time, since there's more nutrients available, our anti-inflammatory cells can be able to become more powerful and stop our inflammatory cells. So it was already shown that in models of inflammatory bowel disease, Zorafen could actually reduce in life the inflammatory cells, and also in cultures, and at the same time increase the amount of anti-inflammatory diplomats. And of course, there's still a lot of work in order to be able to give it to humans, but we are still working on that, and we hope that this compound of us could eventually be able to treat people with autoimmune diseases. So I don't know if people would like to give us more questions or to explain a little bit more about our compound, or perhaps share their feedback on, like, if there's someone that...
Thanks a lot, Maya.
I hope I wasn't too...
No, it was really perfect. Don't worry.
Okay.
You just have to be very relaxed. It's a laid-back Saturday, so make yourself very comfortable.
Yes, yes, yes.
There's nothing
Sometimes
like that.
I was used to trying to force with these 3 minutes usually, because in the street people are coming, going, and they cannot stay longer. But now
because we are trying to also engage people who don't belong from the science background, because usually they really don't know, what a scientist does, and because for them it's always eureka, and that's not usually the case, because we know behind the scenes what exactly is happening.
This is why they should do
it.
more. This is it's nice that we have this event so that people can actually get into.
Okay, so while we're waiting for, actually, I had a question.
Yes.
Because you mentioned about the Zorafen, right?
Yes.
And so, can you mention what are the side effects of Zorafen, for example?
So it had been shown in high concentrations. I mean, it's the dose that makes the poison. First of all, for every kind of either drugs that are produced in a laboratory, or even natural compounds that can be used as medication drugs, it's always about the dose. Zorafen in very small amounts can, in vitro cultures where we took the cells, our immune cells, and try to see the effect of it on them, it was really effective in low concentrations, so the more it might be toxic. However, when someone checks about side effects, don't only checks about toxicity, but also side effects on other cells, and or like the tissue, if it's affected, or... So we did do a screening. As I said, I applied it into in vivo models using mice. I'm sorry if this might make some people feeling uncomfortable with that, but it's one way that we can actually test whether in humans it could be applied. Yeah. So we checked, and it didn't seem to have any negative effects on the quality of life of the treated mice. It affected not only our effector T cells, but other inflammatory cells that play also a role in the fight against pathogens, but to an extent that this wouldn't make our immune system not strong enough to fight infections. So it really improved the disease symptoms. It reduced the symptoms of E. colitis in mice, but it didn't really affect to a great extent the functions of the animals.
So Naya, because we talk so much about, coronavirus these days, right? And then we are always like, what are the drugs coming up? Is it the remdesivir, or is it this, or is it that? And I guess we, as like biologists, we always get questions like, what's the new thing happening? So I don't know if I am right. If I'm not right, just correct me, because even Zorafen was used for, at least in HCC, in hepatocellular, like in the cancer of liver, maybe. but is it also specific for more viral diseases?
I think there was a group actually in our area where we did our PhD in Hanover that were actually trying to use Orofen for antiviral responses. I have to say that I'm not yet very informed about the results, but someone would expect that the compound will act on cells that are starving for fat, because it blocks this desire for fat. So, and I assume that, to some, it depends on the pathogen. So it depends also on the virus and what kind of cells, as you know better as a virologist, it would it will irritate, let's say, in order to, for them to start the fight against it.
Suffer, basically.
Yes,
Cool. It's very good.
But regarding the corona, please don't forget to wear your mask. It's a must. It's the new fashion trend. So,
so Naya, we have one question over here where they mention about how are the compounds screened, you know, like how do they look for this? So, and there's a rider to that are you running lab experiments or is it mainly computer stimulation?
No, it's actually lab work that we do. We had a collaboration with chemists in the Helmholtz Centrum in Braunschweig, where they actually isolated from us the individual compounds from that bacterium from the soil. And we had like pure, in a powder form, the pure products separately. And then we just isolate the cells either from human blood or from mice, and we culture them in the presence of the individual compounds in different concentrations, and in that way we can titrate the effect to see in which concentration and above it might be toxic for the cells, and which is the minimum concentration that you can actually see an effect on the function of the cells without actually killing them. So this is how we select not only compounds that can affect directly the effector T lymphocytes, we can also, we try to find also compounds that could be used as adjuvants for vaccination processes. So that's how we do it. Once we find a compound, like a candidate that is promising, then we dive more into the mechanism, how exactly it affects. For example, Orofen was also known from previous studies from plant biologists and microbiologists, its action, but not in immune cells. It was more known how it affects bacteria. So we also addressed the mechanism on how it affects human cells, and addressing the mechanism then led to us testing whether it also has side effects on the desired cells or on other cell populations that are important for us. And
Okay. And one last one, someone asked about the effector types. Are there different effector types, or depending on the different pathogen, for instance?
The different pathogens, of course, induce different responses. This is a way of our body to have a more specific and strong response towards them. For example, Th17 cells, the cells that I'm more interested in, the effector, the red ones, are for extracellular pathogens or fungi, responsible for eliminating them, and you can find them in great amounts in the intestine. At the same time, you also have the Th1 cells or Th2 that play a role in asthma or lung infections. So each individual cell population is responsible for killing different defending ourselves against different pathogens. Something that someone has to mention, though, is that there is a kind of a flexibility between that. They don't decide a fate 100%. They can still be discussed and change according to the environment slightly. So if there is a demand for a Th1 response, the Th17 cells can also start producing the weapons that the Th1 cells do. Zorafenib, though, had a more generic effect on effector T cells in general and not regulatory. So it makes it a nice candidate to overall affect effector T cells that are implicated in autoimmune diseases, because you usually have Th1 and Th17 effector cells fighting a lot during autoimmune diseases. So yeah, it gives a little bit more wider effect, but it's enough to minimize the inflammation.
Cool. Thanks a lot, Naya. It was
Thank you,
really nice having you here
It was
and
really
sharing
nice
your experience.
to be part of it, and yeah, I look forward for the next sessions.
Yes, sure.
Good.
Thank you.
Bye, people. Thank you very much.
All right. So from an immunologist, what do we move next to? So let's go to the brain. And it's a very interesting talk, actually. So it's by Dr. Manuela Celleto, who's a comparative in the Department of Comparative Psychology at the Institute of Experimental Psychology at the Heinrich Heine University here in Düsseldorf. And her topic is apple or candy. For you or for me, let the brain choose. It's a difficult choice, but I guess Manuela will help us answer this. So Manuela, the stage is yours.
I'm arriving.
Yes, we're waiting.
So, thank you very much for this kind introduction and also for organizing this amazing event. Well, hello, my name is Manuela, and I am a researcher in cognitive neuroscience here at the University of Düsseldorf. What I study has something to do with what I think is the most fascinating organ in our body, which is the brain. If you're not familiar with the brain itself, here I have Rita. She's willing to show us her brain. So you can see half of the brain here and the right half here. I don't know if you can actually see from the camera. And if you're curious to see how it would look like from the outside, here it is. So this would be our brain from the outside. So you have to imagine something pinkish, wrinkly, and also squishy, so it is not hard as this one. Now, as everything in our body, the brain is made of cells, and I need another prop here. So this is only a puppet, but it well serves the idea of what our brain is made of. So this cell is called neuron. We do have different types of neurons in our brain because they have different functions, but this is one of the typical ones. So it has something that is called a body, so this part here. Then this has a long tail, which is called axon. And then there are other branches which basically allow each neuron, so each cell, to communicate with other cells. What is important here for you to know is that these are super, super small, and it is even estimated that we have in our brain up to 100 billion of these cells. So you might imagine that our brain is extremely complex. We cannot, so only one person cannot study everything related to the brain, so all functions that the brain can subserve. To explain you what I study, and what I do is basically look at the brain and understand how us, as humans, make decisions. And to understand that, I'm going to make you an example. So imagine yourself that you are in your afternoon break and you want to have a snack. I'm pretty sure, so you can imagine right now, and I'm pretty sure that some of you might go for a chocolate bar, some other might go for a fruit salad, and others might even go for an energy drink. Maybe you still have work to do, study to do, so you need some energy. What is important here for you to know and also to remember for the rest of the talk is that we all make different decisions. So remember that. Now, I'm going to give you a similar example, but a little bit more specific. Now, imagine that there is a person who is on a diet, and she has to make your very same decision. She is on her afternoon break. She has to decide what to eat. And she has in front of her two options, and I need my props here. So, she has in front of her two things. She can either have some of these candies. They even have a brain shape, so she really loves this. They're her favorite candies. On the other hand, she can have an apple. Now, you might guess that, remember, she's on a diet, right? If she chooses the candies, these are not a very healthy choice because candies have a lot of sugar, or at least more than an apple, and so this will not help her with her future goal of losing weight. Whereas if she goes for the apple, it is definitely better for her diet because in the long term, she will lose weight. Now, you might have heard in the news the word self-control. We can say that if this person, remember, she's on a diet, goes for the candies, this person has low self-control, so she is a bit impulsive because she's giving into temptation of something that is very tasteful right now, but she's losing her long-term goal, so she's forgetting that in the long term she wants to lose weight. On the other hand, if she goes for the apple, yeah, she doesn't get much now, but she will get something in the future, so she is self-controlled. Now, what I do to study this is to have participants, so you have to imagine people, usually my participants are young adults, they come to a special place where there is this large tube. This is called a scanner. You can also imagine to be my participants actually. So you arrive at the lab, you lay down on a bed within this large tube, so within this scanner, and what I'm able to do is to make pictures at the brain of my participants when they are making these choices. So this is a very loud machine, so I usually give earplugs to my participants, so everything is fine. They lay down, they see on this screen a series of choices. To be honest, most of my studies involve money more than food, because it's more easy to study, you know, to make this, to ask decisions with money, but this example well serves the idea. So now imagine a lot of participants, because we never test only one person, and this person never only makes one choice. We do need many participants, and each of these participants has to make a lot of choices. Now, what do I get when these participants are in the scanner? I'm showing you. So, as I anticipated, I make, is there any problem? I make pictures to the brain, and this is how it looks like. So I'm not sure, these are really small, whether you can look from there. So remember the first model that I showed you of the brain. So this looks pretty similar, right? It's like we are looking at the brain from the middle. Whereas this one is a bit more from the lateral surface, so from the outside. This is not what actually a brain looks like. So you have to imagine that this is not one person. This is more person, one over the other. Remember, I have to ask the same questions to many people to understand what's going on in the brain. And most importantly, we do not have in the brain this colored spot. You might have also seen this again in the news or newspapers. I get this colored spot only when I then go to my office. I sit in front of the computer. It might look like, it might sound a bit boring, but actually it's really fun. I do analyze the data that I collected, meaning I look at the behavior of my participants, so the choices of my participants, apple or candies. Are they on a diet or are they not? And also, I look at what was happening in the brain while my participants were making the choice. So the color is basically a way that the computer, let's say that the software, allows me to see where in the brain, what parts in the brain were actually participating, so they were engaged in making the decision. I don't know if I'm able to share with you the feeling that this is really interesting for me. So I'm really excited when I get these pictures, and to be honest, this is also enough for me to do science. However, you might wonder whether this is even useful. I mean, yeah, besides rewarding myself? Is this useful? Why do we need to know what's happening in the brain when participants are choosing apple or candies? Now, if you remember, as I said before, we all make different decisions. This is very important because if I can look at more brains of participants who are making decisions, I'm actually able to see what makes the difference between one person and another. So what is different from this person who is on a diet and can choose the apple as compared to this other person who is on a diet but is choosing the candies. So once we know what is the difference between one brain and another, let's say, so how one brain is functioning and how the other one is functioning, we basically know how to help people who have low self-control to actually be able, let me oversimplify, to choose the apple if they are on a diet. And I might give you an example so that you can actually understand why this could be useful. Researchers before me have found out that if we think about the future, this helps people, let me again oversimplify, choose the apple over the candies. Now, why is that? You might wonder, yeah, that is because they are thinking about the future, right? So they are representing better their future goal, losing weight, and so they're better able to go for the apple and the candies. And this is totally true. However, what you might not know is that the reason for this really lays in the brain. Meaning that when participants are choosing, or when you are choosing actually between an apple and candies, among the several regions that are activated in our brain that makes you make the choice, there is this part here in the front. It has a very long name, but we can call it frontal part of the brain. This part is basically able to take into account a lot of information, like how much do you like the apple? How much do you like the candies? Are you on a diet or you're not? Maybe how hungry are you? Maybe you are on a diet, but right now you are not hungry, so it might still be easy to choose the apple. And many other things, like how far away is the end of the diet, and so on. Now, why thinking about the future helps participants making better, let's say, better choice, or at least go for the apple instead of the candies? Because when we think about the future, among the regions, once again, that are activated so that they're engaged in this action, there is once again the region here in the front. So it's basically like when we ask participants to think about the future, we're basically training this frontal part to better represent all the information that this region needs to make the good decision. Now, I don't know if there are questions. Okay, then I can stop here for now. Thank you. Otherwise, I would go on.
Yeah, you can actually go on as well. So there
no, it's fine.
You can actually go on with some of your pictures, because there was someone who said that probably if you could focus on your picture. Sorry. Keep forgetting this. Sorry. So if you can actually focus on her pictures and if she can explain it better, because I think there were a lot of people who didn't really, you know, can't see, and if you can really like show it, because
yes, of course. So as I said, first of all, this is not one brain. It's more pictures of many of the brains of many participants, one over the other. So this is why you get this smooth image. You don't really get to see very well the structures. And these colors, so they are not in our brain. So this is just a sort of trick, a visual trick, to that the software, that the computer uses to tell me what are these regions in the brain that are participating to this decision. Okay. I don't know if I have to say, for example, what is this activity? What does this activity mean?
So there was a rider to this actually. So someone mentioned that you see these activities only during the scanning or after that?
No, only during the scanner. All so the participant has to be on this bed with this large tube around the head. So the participant has a sort of screen, let's say. She reads the questions and she presses buttons to answer to the questions. And only in that moment the scanner scanner, so this tube is, so this machine is able to make pictures. They are not just the pictures that we make with our smartphones or with our cameras. So these are actually pictures of the activity in the brain. To oversimplify, we might say that what is the activity in the brain? So in our previous talk, we have heard that cells need to eat like us, right? So we need energy, so we need food. So the cells use oxygen. So this machine is somehow able to detect which parts of the brain are using more oxygen. So if they are using more, it means that they are engaged in the action.
Okay, so there is another one just coming in that someone asks, what causes decision paralysis?
Decision paralysis? I'm not an expert of that. I don't know.
All right.
I cannot answer, sorry.
Probably
but I will
really
definitely look at that.
So there is one more that, have you looked at the effect of any drugs, example alcohol, on the brain activity when making decisions?
So not personally, but this is a question very, very related to the kind of decision that I make. Let's assume, where do I have to go? What I study, so this kind of decision between something that I can get right now, so the taste of the candies, and something that I can get only in the future, like losing weight. It has been shown that, always imagine examples that involve money, first of all, and it has been shown that people who are actually addicted to several drugs and also alcohol, they are very bad at making these decisions. So they really cannot, let's say, they are not patient, so they cannot wait for to get a future reward or a future outcome. So they really want something right now. So they are really they are deemed as impulsive.
So actually, because you spoke about the cost, so actually there's something we all wonder, that somewhere we kind of saw that people will pick up something which is free of worse quality, or something of very small quantity but of better quality. Is this true?
Yes, it is true. Once again, this is a different type of decision. This might be related, so it of course involves the brain, and I'm pretty sure, so I've never studied this, but I'm pretty sure it once again involves this the frontal part of the brain, because this is really the part of the brain that gives value to things. So it might be that for some persons it has a bit higher value, something that is free but worse cost. That's something that is expensive but of a better cost. But this, of course, it's not only about a matter of brain, right? It also matters whether you have a high income or not. And these are actually, by the way, all variables that we take into account when we make these decisions. So
one of them.
Exactly. So you might guess that a person who has a high income is much better at following her diet, because she has more like availability of like alternatives, not only just the apple or bad candies.
That's true.
Yeah.
Okay. Cool. Thanks a lot.
Thank you very much.
And it was really nice and very intriguing, and I think there'll be a lot of questions popping in. So if you guys have any questions, we are going to post the handles from Manuela, but also from our previous speakers. So feel free, just drop in your questions anytime during the whole time. They're very friendly scientists. They're not going to give you evil looks. Please feel free really at any point of time to ask them questions, because as I always said, that no question is stupid, and this is now your time to ask the questions. Thanks a lot, Manuela.
Thank you very much.
It's really lovely having you
organizing.
here.
Thank you.
Cool. So actually, we thought that probably we need some food for thought. So I thought maybe of dropping a sort of a science fact. So we talk about science and we talk about Nobel laureates, right? So do you actually know how many Nobel laureates have been there? So just for information, there are around 950 Nobel laureates known, and out of which only 53 are women. And from these 53, only Marie Curie is one who received the Nobel laureate twice. I just thought it was a good food for thought, if you might not know. So some housekeeping instructions for our speakers as well as for our viewers. Please keep popping in your questions at any point of time. We will try to ask all our speakers about your questions, about your interests. And as I said, just you can also type in their Twitter handle, Insta handle, or LinkedIn page, or on the Facebook page, and we will try and answer all your questions. but as of now, it's a Saturday, so just it's a laid-back Saturday, so like sit there and enjoy a fun ride on a soapbox train, I would say. So now we will have a little stop over here because we are trying to get in our next speaker who will join us from Hanover. And there will be a pause for a minute or so, and then we will be back. So we will join her on Zoom.
So my name is Veronica Duran, and I'm a postdoc coming from Colombia. So I have some drawings here to show you. So my country is located here. Sorry, I'm just gonna increase this one moment. Yeah. My country, as you can see from this map, is located in the northern part of South America, and it has access to the Atlantic Ocean and the Pacific Ocean, which makes it a country, a very biodiverse country with lots of flora and fauna, and makes it, yeah, for me, one of the prettiest countries in the world. I come from Bogota, which is the capital of this country, beautiful city on top of the mountains, of the Andes Mountains. And I actually studied microbiology at the University of the Andes, which is one of the, let's say, most renowned universities in Latin America. So I was always fascinated about microbiology and the microscopic world. And this is why I wanted to continue my career. And for this purpose, I came to Germany, as you can see here. So in Germany, I continued my master's studies at the TU Dresden. I did the molecular bioengineering master's program. And then I came to Hanover, to Twinker, which is the institute in which I am at the moment, to pursue my PhD studies. And for my project of PhD was dealing with tuberculosis. So as you may have seen from the program, for this opportunity, I called my project Sugar Spheres to Treat Tuberculosis, the new magic bullet. So first of all, what is tuberculosis? So tuberculosis, it's a disease that is caused by a bacteria called Mycobacterium tuberculosis that you can see here. And this is a disease that is mostly a pulmonary disease. And as you can see, it is transmitted via aerosols from a person that is infected to a person that is healthy. And then in this new healthy person, it will go into the lungs. But why exactly was I so interested, or why did I want to do research for 4 years during a PhD for tuberculosis? So the reason for this is that although not so many people know about it, all good? Okay. Although not so many people know about it, in 2018, 10 million people fell ill with tuberculosis. So we're talking about 10 million people. And among these 10 million, actually, and besides these 10 million, an estimate of approximately 3 million people actually were not diagnosed or reported. And additionally, 1.5 million persons died in 2018 for tuberculosis. And this is especially a big issue in patients that have HIV. So if these are the statistics and these are the values, then you might be asking yourselves, why are we not talking so much about tuberculosis? Or why actually so many people think that this disease is already eradicated and already forgotten? And I believe if you see this map, this is because if you see the countries that it is affecting, it's unfortunately countries that are very, let's say, are like undeveloped countries. So as you can see here, we have South America with a very high incidence, same as Africa, especially southern, like sub-Saharan Africa, and Southeast Asia. These are the places where let's say the majority of cases of tuberculosis are present. And one of the main, let's say, problems with tuberculosis is not the fact that the disease is actually very harsh for the patient, so they really feel all the symptoms that we know, so fever, lots of coughing, and they can eventually die from the disease. But actually the real problem is the treatment. So in order to treat tuberculosis, patients require 180 days of medication, as you can see here. So this means 6 months in which they have to take minimum 4 antibiotics daily. And not only this, but additionally, they have to pursue different exams, such as X-rays, lab tests, and of course, as all these, like with several infectious diseases, they must go on follow-up, and they must test and also control all the contact persons that were included. So to have an estimate of how much or the cost per case of tuberculosis, you can see here that if it's a drug susceptible bacteria, per case it will cost an estimate of $17 US. Whereas if it's a multi-drug resistant that you see in the middle of the pyramid, it will be around $150,000. And if we're talking about extremely drug resistant, we can reach up to $500,000. And the reason for the emergence of these drug resistant strains is because you can imagine if these patients have to take for 6 months 4 different antibiotics, at the end, I actually know from patients that I have seen that they say, okay, I'd rather have the disease than having to take these pills every day, which of course have very side effects. And the problem is that most of these drugs are taken systemically, meaning that they are like ingested via pill and they disseminate throughout the whole body. But the amounts that reach the lung, that is the place where the bacteria is actually residing and causing a disease, are quite low. And this is why the concept of targeted drug delivery arises as, let's say, a potential alternative for the treatment of tuberculosis. Oh, yeah, okay, so what you can see here, this is the approach we're following now. So basically, a patient takes the antibiotics and they are disseminated throughout the whole body. But actually, what we are aiming for is that the patient takes the pill, or like not takes, but inhales, for example, which is what we want to apply, that it's able to inhale the antibiotic. And in this manner, the drug or the antibiotic will reach particularly the lung, meaning that in this way, we can reduce the amount that is needed, because basically we are making it more efficient by bringing it to the site of infection or the site of affection. And also we reduce all the side effects that are associated with these long and complicated treatments. And in order to deal with this targeted drug delivery, then one of the main ways to do this is by using nanoparticles or nanomedicine. And as the name says it, basically nanoparticles are particles that can carry, in this case, antibiotics that are in the nanoscale. So when we talk about the nanoscale, if you see this here, I should have mirrored the video, but I'm so sorry. If we start here from the beginning, a molecule of glucose has around 10 to the minus 1 nanometers. Then a protein, it's a bit bigger. Then we find here the DNA. Then we find here some viruses. And here we find bacteria, like, for example, Mycobacterium tuberculosis. Furthermore, increasing for cells, this is the size of a salt grain, and finally reaching to a tennis ball. And particularly in this range here is where we find our nanoparticles. And in this particular, so for my project, what we used, I don't know how to put this, okay, maybe like this. What we used is that we used spheres that were coated with the sugars coming from tuberculosis. So you see this blue symbol here represents sugars that are on the outer part of the bacterium. And then we had spheres here made of lipids that were covered with such sugars. And on the inside, they had antibiotics. So basically, this is also called like a Trojan horse approach, because what we were aiming for is to mimic the pathway that the bacteria, Mycobacterium tuberculosis, uses in order to reach the lung. And we just tried to mimic this, but instead of it being a bacteria, we tried to use spheres that are made of lipids, which is something we can take, let's say, easily and would not activate our immune system or wouldn't cause any adverse effect. And these lipid spheres were loaded with antibiotics. In this case, we tried it with levofloxacin, which is an antibiotic that is given to people once they start presenting this drug resistance. But to understand why this is such a good approach, we need to understand how is the bacteria actually causing a pathogenesis. So in our immune system, we have some cells that I really love that are called macrophages. So you see them here. So actually macrophages, the word macrophage is derived from the Greek makros, which means large, and phagein, which means to eat. So what you see here is basically a macrophage that is eating a bacteria. And as I just said, the bacteria is full of sugars, and who doesn't want to eat sugars? And this is why macrophages will eat directly. So we have lots of macrophages that are sitting in the outer part of the lung. And then the moment that we inhale either the bacteria covered with sugars or either our spheres also covered with sugars, these macrophages will immediately try to eat this up. But the thing that happens with tuberculosis is that this is actually a very, if we can say, a very smart bacteria, because what it does is that it enters the cell, and usually what should happen is that the cell would be able to degrade. I hope you're able to see it here, but then the cell has inside these compartments that start getting very acidic, basically, that they can then like break down the bacteria into pieces, and then, as you see here, then release it into little pieces that will then serve as signals to start an immune response. This is how it should work normally. But the bacteria, as I just said, she finds a way to manipulate this whole system and this whole mechanism, and she just stays hiding in these little bubbles that you see here. And this is actually what makes it, this is actually one of the reasons why it is so hard to treat tuberculosis, because basically you have to target bacteria that are hiding within your own cells of the immune system. So not only it's hard for them, not only it's hard for our system to, for our immune system to start a response towards them, but it's also very hard for the antibiotics to actually reach these places. And of course, this would mean that lots of these cells, macrophages and neighboring cells, would have to be killed in order to kill the bacteria as well. And this is from where all the side effects come. And this is why then the trick that we are applying can be resumed in this scheme that I hope you can see here. So now I won't show my face so that you see this. So if we have the bacteria, like this, if we have the bacteria entering this, if you imagine this is a macrophage, yeah, this is the inside of a macrophage. And if we have the bacteria that is covered with sugars, then these sugars will be recognized by specific receptors. Then they will be internalized and they go and hide into this bubble. That is the one that I showed you on the image before. Then if we come on the other hand with our sugar spheres, then they will be recognized by the same receptor and basically enter a very similar pathway. But the thing is that these sugar spheres do not have the capacity to, let's say, block this procedure. So in the end, what will happen is that they will fuse together with the place where the bacteria was hiding. And once they are hiding there, these spheres won't resist the acidic pH, won't resist all these proteins that are trying to chop everything into pieces. Then the sphere will be chopped down and all the antibiotic will be released, therefore killing the bacteria. And this is why we call this the magic bullet. And this is actually what I worked during my PhD thesis, which I finished actually 1 month ago. And we could actually prove by infecting cells. So we isolated cells from human lungs. So people, for example, that were undergoing lung transplantations, whenever the lung was removed, then we were able to get this lung tissue. Then we isolated these macrophages, so these eating cells, and then we infected them with mycobacteria. And what we try to do is we try to treat or like clear the infection on these cells, either by applying antibiotic that was soluble, so as the current therapy is, which could correlate to taking it systemically, versus the same amount of antibiotic, but just encapsulated in these sugar spheres. And we could prove that actually by only encapsulating it in these spheres, we were able to reduce the bacterial infection in very high amounts, and still the cytotoxic effect and all the side effects were lowered. So of course, now we are hoping to go for the next steps of this, let's say this was the preclinical study, but of course now we are trying to go for the next steps that are trying to see if these spheres can be then inhaled, and in this manner then really reach the lung and then be able to treat not only the cells that are infected, but also a patient that is infected. And yeah, I think with this, I'm done. I don't know why I was so fast. Because yeah, I think with all the technical issues at the beginning, I got a bit nervous, but then this gives us more time for questions. So I will see them here writing in the chat. Yeah, they're saying thank you. I thank you girls and guys for the organization. Question for you, okay? Is the size of the sugar sphere important? This is a very good question. And yes, it is very important, because depending on the size of the spheres, then we can either go and let's say take the same pathway as the bacteria, as we're seeing here. You see that they take the same pathway. And this size has to be between 100 and 200 nanometers. But the moment that we either increase, for example, increase the size of these spheres, then they won't be able to go anymore via the same pathway. And then we don't reach the same, let's say, bubble inside of the cell where the bacteria is hiding. So this is actually a very important issue and one of the, let's say, main challenges when working with nanoparticles, because these nanoparticles are made also in a way, like in a spontaneous way. So as they are made of lipids, you basically just put a lipid solution in a water solution, and then just by hydrophobic forces and entropic forces, they will make these spheres. So then really calculating that all of them have the size that works for this approach, it's challenging. So you really have to filter them several times in order to reach for the, let's say, the optimal size. But yeah, this is very important, and it's a very good question. And now I have another one. It says, has nanoparticle use or approved for any other diseases known? So yeah. So actually, there are already nanoparticles that are approved and that are used for clinical trials. And what can be, let's say, of relevance at the moment is that you have heard, many of you might have heard, that companies like CureVac and Moderna and many other companies are developing vaccines for the coronavirus. And actually, these vaccines that are, let's say, taking the lead at the moment for coronaviruses are based on lipid nanoparticles as well. So basically, particles that are having lipids, similar as in this approach. Just the difference is that now, as we want to treat tuberculosis, we loaded antibiotics. But in the case of working for a vaccine for coronaviruses, then they loaded them with a specific molecule or specific, let's say, we can call it antigen, not to make it complicated, so that then this can enhance an immune response towards the coronaviruses. And these are actually some of the vaccines that are taking the lead at the moment, and all of them are based on nanoparticles. So I think I have no more questions. So if some other viewers have any other question, then they can come back to me either on Facebook, on Instagram. And I thank you all then for this nice opportunity and also for letting me join virtually. I'm sorry for all the technical difficulties, but I hope you enjoyed it. And then let's keep on listening to the other speakers.
We're back. I hope with some technical difficulties we were still able to get Veronica streamed from Hanover, and I really wish that you guys would have liked her talk. And if you have any questions, we will put her Instagram or Facebook handle, and you can just drop in a question and ask her questions at any point of time. I'm pretty sure she will try and answer all kind of questions. So without any further delay, I will hand over the mic to my colleague Sepi, who will take over the German part of this session.
Thank you everyone for joining our Soapbox Science event in Düsseldorf. It's our first time. We had some technical difficulties, but we hope we could still convince you all to have a nice Saturday afternoon. So I'll start from the end. Maybe we can all present, say something about ourselves, what we do, and yeah.
I'm Cristina Sarasua, and I'm doing my PhD in volcanology.
I'm Catherine McMillan. I was the main organizer for this event, and I wouldn't have been able to do it without these wonderful people here, so I have to thank them, and of course our fantastic speakers who I really hope you've enjoyed listening to. So I'm a physicist, should probably have said that, and I work at eine Universität.
My name is Sepide Sadat. I'm a psychologist and neuroscientist by training. At the moment, I work as a clinical neuropsychologist, so putting all the knowledge that you learn at university into practice. And yeah, it was a great pleasure working with these ladies and really promoting the visibility of women in science. And we brought out the main mission of this project with our great speakers.
And I'm Audrey Nguve. I'm a biologist working at the Heinrich Heine University as well in the area of stem cell research. And yes, I would also say, join the ladies and say it was a great talk afternoon, and we enjoyed all the talks, and then everyone hopefully did learn something from this. And hopefully we're also able to convince a few young ladies to maybe join into the science world.
Yes. All right, and finally it's me. I was moderating the English session, so I'm Tanvi here again. I'm a virologist. I mainly work in the field of liver immunology and virology, with the focus of hepatitis diseases, so viral hepatitis. And it was really fun being a part of this great dynamic team, and there are faces from different parts of the world, but we connected well, and that was, I think, because of the core reason and the passion for science communication and also for the promotion of women in science. And I think it was really work well done, and I hope that you guys would also appreciate it. Thanks a lot.
Thank you, guys.
And last but not least, we'd like to thank our sponsors and our supporters, in particular Heinrich Heine University. We'd like to thank also Surplus, also from the Heinrich Heine University, and Kofenster, who helped us compile the speaker videos in the beginning for the introduction round for the speakers, and also the Bürger Universität from the Heinrich Heine University in Düsseldorf. And with that, we'd like to wish you all a pleasant Saturday afternoon, and remember, you can always come back and watch our videos on YouTube and ask your questions.
Like and share.
Like and share. Thank you very much.
Thank you.
And before I forget, also our camera crew, we'd like to thank them. We come. Yes.
More important.
Thank you very much. Have a lovely afternoon.
Yeah. Thank you, guys. Bye-bye.